All right, let's talk a little bit more about the kidney and some reciprocal induction of said kidney. So this is kind of just a bit of an overlay or overview of reciprocal induction. We'll kind of go through the process of it here. Let's get my thing here. Okay, so we see we have this metanephric mesenchyme here, and then we have our ureteric bud that's kind of growing into that direction, right? And so it'll grow actually into this metanephric mesenchyme area or mass of cells. And because of that proximity or contact with the metanephric mesenchyme, it causes this bud, the ureteric bud, to branch. So you can see that we started with a single branch here. It contacts the metanephric mesenchyme. It causes it to start to branch, and as it grows, it continuously will branch more and more. So at the tips of the branches, the epithelium induces the mesenchyme to aggregate and cavitate, as we can kind of see this process here. Where we go? To form renal tubes and the glomeruli, and that's where the blood from the atrial is filtered in those glomeruli. So you can see that what's happening here is the mesenchyme. This is why it's called reciprocal induction. So the mesenchyme is instructing the ureteric bud to branch, and then this growing bud is instructing the mesenchyme cells in return to start to cavitate and form these renal tubes and glomeruli. So you can see at stage here we get the pretubular renal aggregate. So it's just a collection of what's going to be kidney cells kind of aggregating around the tube here, and start to kind of lobe off, cavitate, and form these inner cellular areas here. We get this connection that's occurring near with the collecting tube here, where this renal aggregate actually starts physically forming one, we should say, with the what used to be ureteric bud at this region. We're at this early stage, the ureteric bud. So you can see the kidney then starts to form. We have the glomeruli down here in the arterioles where the blood is filtered, and this is Bowman's capsule we're not going to talk very much about. Okay, so let me double check my notes. So when this mesenchyme has condensed into an epithelium, it digests. Okay, so when the mesenchyme aggregates it like this and condenses into this epithelium, it digests the basal lamina of the ureteric bud cells that induced it, and connects to the bud epithelium. So that's kind of around between these two stages right here. So we see that once they start to form this epithelium and kind of close off from the surrounding tissue, it'll digest that basal lamina and just kind of straight up connect with it. And then a portion of the aggregated mesenchyme, this pretubule condensate, let's talk about these cells here, a portion of that becomes the nephron. So that's the renal tubes and Bowman's capsule over here. While the ureteric bud becomes the collecting duct for urine. So that's kind of in a nutshell what's happening here. So we're going to look a little bit closer at some of this stuff. So this is just kind of an overview of kidney branching. So this is a kidney rudiment from 11.5 or 12-day mouse embryo. So this mouse had transgenic GFP fused to HAX-B7, which is specific for these renal tubes. It expressed green fluorescent protein in the Wolffian duct, which is this big duct right here, and the ureteric buds. The cool thing about GFP is since it's living tissue, since they cultured it, it's living tissue, you can kind of watch it grow. It's all one section. It's not multiple mice. It's just this one's continuing to grow. It's the same sample each hours. Okay. So what's also kind of cool is that since we know so much about this this process, we can induce some of this tissue formation from pluripotent stem cells. So we have this schematic mechanism of generating this ureteric bud and the metanephric mesoderm from these where is it, from the posterior from the posterior mesodermal precursor cells. Posterior mesodermal precursor. Okay, here. So we have the posterior here and the anterior, and this is over time. These posterior, the light pink ones, posterior mesodermal precursor cells, they are migrating from the posterior mesoderm early during gastrulation, and they leave this high WNT area, and they proceed towards areas of more FGFs and retinoic acid. So when they do this, these become progenitors for the ureteric bud epithelium. So it's all about, remember this bud is growing and moving, and these cells are kind of reorganizing at this time. And so when they leave their posterior position where there's high WNTs and they move towards these FGF and retinoic acid areas, they become progenitors for this metanephric mesenchyme up here. Or if the more they have, they can proceed even further and become ureteric bud epithelium, right? So sometimes it's not the gradients that come to them, it's the cells that move through the gradients, if that makes sense. All right, let's move on here. So here, this is just kind of, you see we have the notochord, the somites, the presomatic mesoderm down here, which would have the high W, or in this case has high CYP 26, which is a repressor of retinoic acid. So once these cells start to migrate, that retinoic acid is not in this area because CYP 26 is repressing it. But further back, more anteriorly, we have higher retinoic acid, which can cause them to differentiate. This is just an immunofluorescent analysis of a kidney organoid. And so this was where they took induced pluripotent stem cells and they exposed it to WNT and then FGF, and then kind of saw how this cascade proceeded. So let's move on. Let's see. There's no insert on this one. So this is just kind of showing the different differentiation of these cells. Just kind of cool image. Not much to take away from that one. Should have taken that one out. So this ureteric bud growth is highly dependent on this molecule called GDNF and its receptors. And so in this case here, we had mice that had retinol deficient cells, and these are these green cells, or retinoic acid, excuse me, these green cells, and RET expressing cells, which are the blue cells here. And when that happens, you can see that the buds, the blue cells, are what migrate and are the ones expressing this retinoic acid or this in GDNF, and those are what cause or are leading the way, so to speak, in this bud formation. So those expressing cells are what cause these buds. And so here is buds from an 11.5 day wild-type mouse, and you can see this. So this is kind of our control for this this GDNF roll, the GDNF roll. So this is just normal. We have our ureteric bud, and we have our nephric duct here. And so in this case, in C here, we have a heterozygous mutation. So obviously, if you're a diploid like human, you have 2 copies of each gene. So GDNF, if you have 2 functional copies of GDNF, this is what your kidney looks like with your ureteric bud and your nephric duct, and you can see the branching occurring inside the kidney. If you have a not complete, a heterozygous mutation, so you're not homozygous for it, so it's obviously incomplete dominance, if you remember your genetic terms, where if you have 1 functional copy, it's enough, but you show symptoms, is kind of like sickle cell, versus 2 functional copies in your wild type. So this is kind of this sickle cell-esque incomplete dominance of the nephric. It's not associated. I don't want you to get confused. It's not associated with sickle cell anemia, but sickle cell is also not incomplete dominance, right? So if you have 1 mutation, you have half functionality. Same thing here. So GDNF, if you have 1 mutant copy or 1 broken copy, you get partial completeness of the phenotype, or partial functional of the phenotype. And if you have 2 mutant copies, you get this. So you get no budding whatsoever. And you get this nephric duct where both copies of the GDNF gene aren't there, and so you don't get this bud formation to occur. Okay, hope that wasn't confusing. I was trying to make a good analogy because some people would probably be familiar with how sickle cell trait and sickle cell disease work. It's not associated with it, but it's also a codominance or incomplete dominance trait like that. Okay, moving on. So GDNF, so we're going to look, here's more GDNF branching of the epithelium. So in this case, the ureteric bud and its branches are stained orange. It's kind of gold, I would say, but this is orange here. And so this is that cytokeratin stain, immunostain, cytokeratin 18, while the nephrins are stained green with an antibody. And then, so this is 13 days. And then here we have a similar kidney where we have a incomplete or single mutation, right, in one of the genes. So you can see again, the formation is not as clear, or as, I don't want to say clear, patterned correctly. There's less branching. So this is, again, this is the mutated one. I'll go back. This is the wild type. You can see reduced branching, less green area. I am not sure what they're circling here. Didn't put that on there. Okay, so moral of this story so far: GDNF, not incomplete dominance. So 1 copy makes half of what you need, right? A lot of places, a lot of genes are complete dominance, where as long as you have 1 functional copy, then you're good. In this case, if you have 1 functional copy, you're not wild type, but you're not the worst off. You're in between, right? If you have 2 mutated copies, then you just get no functionality, right? And we get that straight line in that. Okay. And this is just 2 different views of that. Now, we also have WNTs again. WNTs coming up over and over, right? Sonic Hedgehog WNTs, FGFs, those are TGF betas. So WNTs, like everything else, play a role in kidney development as well. So this is an 11-day-old mouse kidney where WNT9B here is found in the stalks of the ureteric bud, and WNT11 here, circle it up there, is partitioned in the metanephric mesoderm, mesenchyme, the metanephric mesenchyme, and causes it to condense. That WNT11 will then partition the metanephric mesoderm and induce branching in this bud. So you can see that we have the WNT9B is doing this branching here, and then they kind of show you where the ureteric bud is going to be, and then they also show you, if you stay in WNT, that you can see it would be here and here. So this WNT11 is important for the budding, ureteric bud formation and the eventual budding, while WNT9B, a different copy or version of WNT, is what is important for the expression in the mesonephric duct. Cool. All right, let's look at some more WNT. So you see that a lot of the ways, similar to the Drosophila stuff that we talked about earlier in the quarter, some of the ways to figure out the functionality or what's associated with developing systems like this is to mutate and knock things out, and then look at anatomically what's changed, right? And so a lot of the mechanisms aren't fully deduced in some of this stuff. And so here, an example would be this WNT9B. The minus means that both copies are mutated or knocked out. And so we have wild type. You see full kidney development here. Adrenal glands and the uterus is all the same. So this is in a female mouse. Male uterus, not uterus. The uterus are the same. This is a male mouse. I was wondering, why does my note say male mouse and uterus? Ureters. I am just having a hard time talking. Ureters, adrenal glands, and kidneys are all properly functioning in this image here. Now, in a mouse where they knocked out WNT9B, both copies, you see that we have really... the kidneys are absent, actually, so you don't even see them. We have the adrenal glands there, but WNT9B is really important for kidney development. Obviously, that if you knock out both copies, you get no kidneys. All right, so an important part of the urinary system for you aspiring urologists, this is going to be news to you. An important part is the bladder. No. Anyways, so we talked about the kidneys. Lower down the urinary tract, we get the development of the bladder. And this is connected via the ureter, not the uterus, as my failing eyes was stating earlier. The cloaca, get my thing here. So this originates as the endodermal collecting area for the urine, and then will open up later in development into the allantois. Here we can kind of look at this process in real life. So this is kind of like I don't know if this kind of just shows you proximity to everything. The nephric duct, and then we had the ureteric duct here, where that grew into that mesenchyme that is causing this metanephrogenic tissue to form, right? This reciprocal induction is happening right here. We have the cloaca, which is a temporary organ that's gathering urine at this point. We're not going to get too much more into the genital development, just not enough time. And then the allantois. All right. Okay, make sure I cover everything. All right, so we're going to look at some of these kind of micrographs of how this works. So here, this is a 10-day mouse with a urogenital tract here. We have the nephric duct is stained with GFP, which is that HOXB7 again, which is what stains this, is specific to expression to this nephric duct. So once we get to an 11-day embryo, you can see that we have the ureter and we have this reciprocal induction happening up here in the mesenchyme that's causing this this ureteric bud to start forming this kidney with those condensed or coalesced cells from the mesenchyme. Here we have at this proceeding further. This is a 12-day embryo. And so we have the urogenital sinus down here where the bladder is going to be. We have the ureter and the nephric duct up at the top. And then the ureter separates from the nephric duct and forms a separate opening into the bladder here. And the urogenital sinus developing down at the bottom. That is down at this down at the bottom here. All right. I'm hesitating because I'm thinking, was it important to really cover this? All right, that's it for this this unit. Maybe I'm starting to check out at the end of the quarter a little bit here with my pre-screening of some of these slides. I should have a couple of them. I was like, we didn't need to cover that. Anyways, so that's reciprocal endowment. So moral of the story. This since I was kind of hemming and hawing a little bit. This induction, this is mostly about that reciprocal induction and the players that were important. So retinoic acid, that GDNF, and how that leads to bud development. And if you remove GDNF, you end up getting this not formation of the proper formation of the branching within the kidneys, right? So you get reduced formation. 2 copies is required for wild type, 1 copy for an intermediate phenotype, 0 copies means no branching. WNTs are very important. WNT9B for this duct development, and then WNT11 for the kidney development. But they're also dependent on one another. What's interesting, remember, so here, just to add something real quick, we see 9B is in this in the duct, but it also plays a role in the kidney development, right? So WNT9 or WNT11 is expressed in the kidney, but remember the next slide, if we get rid of 9B, this is the one that normally is in the duct, you just get no kidneys. So while it's expressed mostly in the kidneys, or mostly in the ducts, this WNT9B, it's still important for the kidney as well. Yeah, so that's kind of the moral story. Kind of clear things up a little bit. Reciprocal induction and how it works. So the mesenchyme telling the buds to branch, the branching buds telling the mesenchyme to turn into a kidney. Okay, that's it. I'm going to stop there. I'm rambling. 22 minutes has been a little bit of a longer one. Thank you for watching. I hope you enjoyed. I think we're going to move on to the heart next, because this is also part of kidney development, or the same precursor tissue. So we'll move on to that. Thanks for watching, and I'll catch you in the next one.